Hello everyone and welcome to your screencast on the endocrine system. This is covering both chapters 17 and 18. You'll see that chapter 18 is involved in a little bit more of the physiology, whereas chapter 18 we will talk about the endocrine glands. This screencast is going to be broken up into 6 different bite-sized screencasts so that it's a little bit easier to digest. So the first thing that we'll talk about are some of the principles or more of an overview of some of the chemical communication that takes place within our body. We have 4 major classes of chemical messengers. The first one are our autocrine chemical messengers. So notice that the word auto is in that term, and that means self. So the cell is going to secrete its own chemical messengers, which will then go and have a local effect on itself to release more chemical messengers. Next, we have our paracrine chemical messengers, para meaning surrounding. So these are going to be cells that will release chemicals within their surrounding tissues without being transported into circulation, bypassing that and moving within their surrounding tissue cells. So an example of this would be somatostatin, which we'll discuss in a couple slides. Third, we have our neurotransmitters. These are produced by the neurons. That makes sense because we have the word neuro within this term. And it gets secreted within a extracellular space by our presynaptic nerve terminal. So we haven't quite talked about a nerve cell, but you may have covered in an introductory biology course that are cell bodies over here with extending dendrites, and then we have our axon in which the signal would move through, and at the very end we have our nerve terminal or axon terminal. So we call this presynaptic nerve because it is before the synapse takes place, which would be between one nerve, the first nerve, and the second nerve. So this would be our synapse, and it travels a short distance over to usually another neuron. So we would call this the postsynaptic neuron over here. So an example of this would be the neurotransmitter acetylcholine that could either be transferred to another neuron, or we'll discuss this next quarter, it can be sent over to a neuromuscular junction to skeletal muscle. And lastly, we have endocrine chemical messengers. This is a intercellular type of signal. It's produced by endocrine cells, and all of their chemical messengers will be secreted into the circulatory system. So it's put into the blood and then sent over to, could be very distant regions from where the chemical messenger was first secreted, and then it will have its effect on those target cells that were found further away. So we'll have lots of examples of these endocrine chemical messengers coming up. Now I wanted to do a quick little overview of the differences between the endocrine and the nervous system. They both function to communicate throughout our body, but there are very noticeable differences between the two. So one of the main differences is how quickly the activity takes place. Within our endocrine system, it takes time to secrete those hormones, have them travel through the bloodstream, get over to their target cells, attach to their receptors, and cause the cascade of the result that they're intending. Whereas our nervous system, their activity is very rapid. Within milliseconds, that impulse is sent through the nerve and it gets over to its target organ to cause its result activity. If we go back to our endocrine system, the response here, or the result of the hormone making contact with the targeted cell, is much longer. So for example, we are going to talk about the hormone ADH, and once that is released, it creates these aquaporins, which are water channels within the tubules in the kidney to reabsorb water. And so that response will last a lot longer. We'll be able to reabsorb water a lot longer than if we were to send a signal within our nervous system. So let's just say we send over a motor impulse to our biceps brachii, it contracts, and we're done. The other difference is the type of communication that we're going to have within each of these systems. So in our endocrine system, we have hormones traveling in blood, whereas in our nervous system, we are going to have the nerve impulses travel through the nerves. Now, if we compare these systems as far as the signals they're sending, we could say that it is amplitude versus frequency. So if you look at the left of your screen, we have this wonderful diagram showing you the concentration of hormones within our blood, and we can vary this based on the amplitude. So we can have a lower concentration of hormones, which would result in a weak signal. We increase that concentration, then we get a strong signal, and if we further increase that concentration, then we get a stronger signal. Whereas your nervous system is more of a frequency modulated system. So every signal that is sent is the same. It's kind of all or none. The difference is how frequently we are sending that signal. So here, where it says weak signal, we have only 4 signals here, and they give a little bit more time in between each signal being sent. Whereas where it says strong signal, we have more frequency within the signals that are being sent, and so that increases to a strong signal. And then if we increase the frequency even more here, we get a stronger signal. So let's start with an overview of our endocrine system here. A lot of organs within our body are going to secrete both hormones and exocrine products. So whenever you think endocrine, you should think hormones. When you hear exocrine, you should think ducts. So here's an example of some of the ducts that we discussed within our integumentary system last quarter. And these are always going to secrete components within these ducts and move up to a pore that'll excrete the products. Within our endocrine system, we said that this can take place in a variety of ways, but typically we're going to be discussing a cell secreting a chemical messenger, it going into our circulatory system, and then arriving at its target tissue. Our hypothalamus within the brain has both neural and endocrine functions. So here's a nice little image of that hypothalamus, and you could see the neural tissue here. Some of the axons cascade on down to what we will soon know as the pituitary gland, but we also have blood vessels in here in which some of the hormones that are synthesized will be secreted into that circulatory system so that it has more of a endocrine function. So we'll talk more about that in chapter 18. And then our other tissues and organs that are going to produce hormones would include our adipose cells, thymus, cells within the walls of our small intestine, stomach, kidney, and heart. So before we jump into the hormones and how they are going to get into the cells, we have to talk about how these hormones are released by a cell and how they travel through the blood. So let's first take a look at this cell here that is secreting a water-soluble hormone. This hormone enters into circulation, and typically it can travel through the circulatory system with ease, but other times it is going to need to bind to a protein in order to make it through and last within circulation. Water-soluble hormones can degrade quickly within the hydrolytic enzymes within our blood, and so that's why sometimes they are going to need to bind to a binding protein to make it last and kind of protect it from the hydrolytic enzymes. It can also help protect it from being filtered from our blood by the kidneys. So either way, if it is a free hormone moving within circulation or bound to a protein, it will go through the capillary wall and make a attachment over to a membrane-bound receptor. Now, this is a difference between our water-soluble hormones and our lipid-soluble hormones, which we'll discuss in a minute, is that it has to bind to a membrane-bound receptor. Now let's take a look at our lipid-soluble hormones. You could see that the cell has released it and put it into circulation, and at that point it binds to a binding protein. So we can use an example such as testosterone binding to a very cleverly named protein called the testosterone binding globulin. So they bind together, and once again, this is going to help to make sure this hormone stays protected and lasts longer within circulation. Usually our lipid-soluble hormones have poor solubility within our blood plasma, and that's why typically they are going to need a binding protein. The binding protein will help to make it more soluble within the plasma. And again, it will protect it from being filtered from our blood by the kidneys. So then it travels over to its target cell. At that point, that lipid soluble hormone is ejected from the binding protein and moves straight through the plasma membrane here. It does not need a membrane-bound receptor like we saw with these water-soluble hormones. It moves directly into the cell and can go over to our nucleus and make all of the cellular changes it needs to in here. So now we get into our chemical nature of our hormones. Like we mentioned, we have two major classifications for hormones. We have water-soluble hormones or lipid-soluble hormones. You may also hear water-soluble hormones being called non-lipid soluble hormones. So examples or subclasses of these would be your amines. So these are modified amino acids. You do not have to know the structural composition of each of these hormones, but I just put it up here so you could see that their variations are very slight, but also makes them different. So we have norepinephrine and epinephrine over here. Next classification is our peptides, which would include our antidiuretic hormone that I mentioned earlier, or ADH, and each of these are representing different amino acids that have come together to create this peptide chain. We also have oxytocin. These two are actually released by the posterior pituitary gland, which we will get into in chapter 18. Other examples of this would include insulin or the thyrotropin releasing hormone that comes from the hypothalamus. So as you can see, they are just short chains of amino acids. Next, we have our proteins. So these are larger molecules consisting of a lot of chains of amino acids or polypeptides coming together, being folded correctly, and getting to that quaternary protein structure that we discussed back in Anatomy 1. So examples of this would be your parathyroid hormone or your growth hormone or thyroid stimulating hormone. Now let's get into our lipid-soluble hormones. This would include steroid hormones. So a lot of these are derived from the chemical composition of cholesterol. So examples of that would be cortisol, or a lot of our reproductive hormones that we know, like testosterone, estrogen, progesterone, and it also consists of aldosterone and other adrenocortical hormones. The other type we have are amino acid derivatives. Whoops, I thought I had a picture of that. I guess I don't. And the only one that we would have is the thyroid hormone. So that would be thyroxine, or that's also known as T4. And lastly, we have fatty acid derivatives, and the only one we have to know there is prostaglandins. So now let's get into some patterns of hormone secretion. We can have chronic hormone secretion, which means that the level of hormones within our blood is going to stay relatively constant. So an example of this would be our thyroid hormone that is basically setting our basal metabolic rate within our body, within our cells. The other type we can have is acute hormone secretion, where we are going to have secretion based on the signals that we are getting. The example here is epinephrine in response to stress. So we get a stimulus here. Whoop, there's my cursor. A stimulus here. We have, an anatomy exam coming up. We're stressed out. So we are going to secrete a lot more epinephrine than as we would, let's say, maybe it's after the exam, but we still have to head into lab to study some structures, but we have a pharmacology exam coming up, so we stimulate just a little bit of stress building up, a little bit of secretion of epinephrine, and then we go back. And of course, you guys have an exam the week after too. So, so we get that stimulus again, and epinephrine is released. So it's not something that is very consistent. It can fluctuate. And we have another example down here using insulin as your example of being the hormone that is released. The other type we can have is episodic or cyclic hormone secretion, and an example of this would be your female reproductive hormones. So that time of the month comes around, and we are going to have certain hormones released. Or an example would be before you ovulate, you get a surge of the luteinizing hormone that is secreted, and then we get ejection of the egg from the ovary, and then the LH levels or luteinizing hormone levels will come back down. So it's very cyclic in that way that certain time within a month, these hormones will be secreted.